Thank you for joining the Once Changing the World, which is India's first Future Tech Meets Sustainability podcast. And today, I'm delighted and honored to have with me Professor Isaac Chen, who's a neurosurgeon at the Presbyterian Medical Center of Philadelphia and the Veterans Administration Medical Center. He heads the Chen Laboratory, which works at developing novel methods for restoring the function of the brain after it has been damaged by combining aspects of stem cell biology, neural tissues engineering, and neural interface technologies. Professor Chen is the member of Center for Brain Injury and Repair, Institute of Regenerative Medicine, Institute for Translational Medicine and Therapeutics, and Center for Neuroengineering and Therapeutics. So, Professor, it's a complete pleasure and honor to have you on the show. I'm going to directly I'm just going to directly jump dive into the news, which I just read about, the brain organoids. So before we get into, how you transplanted a neuron into a rat brain, we'll appreciate if you kind of talk about what is a brain organoid first and how do you create it?
Yeah. So the current version of brain organoids started in the early 2010s. And basically, the idea is that we're able to create a tissue in the laboratory from stem cells that mimics what the real organ looks like. So organoids are not limited to just brain organoids. You can make lung organoids or intestinal organoids or kidney organoids. And all these different organoid technologies are based off of the same idea, the same organizing principle, which is that you can take a cluster of stem cells. These are pluripotent stem cells, meaning that they can become any different type of cell in the body. And you can feed these stem cells a recipe. And that recipe instructs the stem cells to become a type of cell and to become a specific type of tissue, much in the same way that each of us developed in our mother's womb. We all started as one cell, an egg and a sperm coming together, and that cell divided many, many, many times to create all sorts of different tissues. And over the years, through developmental neurobiology, many of the signals that control how these different tissues develop in our bodies have been figured out by scientists. And so we can use that knowledge to create these recipes to instruct stem cells in a lab to become similar to certain types of tissue. They're not perfect replicas, obviously, but they get pretty darn close.
So you're saying you take a stem cell, turn it into a pluripotent stem cell, put it in a petri dish, instruct it to kind of, grow, and then it becomes these organs, and it's not restricted to just the brain. It could turn into anything, a kidney, a lung, or whatever. When you say you're building a brain organoid, we're sticking to brain organoid at this point in time. If you're doing that, how close it is to a brain? What's the neuron size? What's the how functional is it?
I think it is important to emphasize that these organoids are not replicas of the actual organ. There's still a lot of work that needs to be done to refine the technology of creating these organoids. That being said, like I said, there are many aspects of these brain organoids that come very close to what the brain looks like. In talking about brain organoids, there's kind of two categories. The first category are what we call whole brain organoids. These organoids are organoids that have features of different parts of the brain all together at once. So, they have some parts that resemble what we call the cerebral cortex, which is one of the largest parts of the human brain, as well as other parts like the cerebellum in the back of the brain and the hippocampus and all sorts of other features of the brain. They're all a little bit disorganized, but you have those different groups of cells. The other group of organoids are called region-specific organoids. And so in this case, you are creating an organoid just resembling one part of the brain. So you can have a cortical organoid that looks like the cerebral cortex. You can have a hippocampal organoid that looks like the hippocampus. You have a cerebellar organoid that replicates the cerebellum. Now, all of these are relatively small. we're talking about something on the order of a couple of millimeters right now. All right. And there are various reasons for that. But when you look at these organoids, what's really, really interesting is that the types of neurons that develop are appropriate for that particular, specifically talk about region-specific organoids. The cell types are appropriate for that part of the brain, and you start to see some sort of structure develop. That structure, again, is reminiscent of what we see in the brain. So as an example, for cortical organoids, the cerebral cortex, what students might learn in basic neurobiology is it has 6 different layers. And the cells in each of those 6 different layers have different functions in terms of how they're connected with other parts of the brain, other parts of the cortex. And they develop in this particularly interesting way where it's called inside-out development. You develop the lower layers first, and then the upper layers kind of develop later and crawl past the lower layers to create the upper layers. And so you see that type of organization start to develop in cortical organoids. We start to see rudimentary layers. The layers are generated in the right timeline in terms of the lower layers being formed first and the upper layers forming later, and kind of this upper layer cells crawling past the lower layer. So all these different features, you start to see that in these organoids. And in my mind, organoids recreate the brain better than any other technology that's currently available in the lab. And that's why there's such interesting things to study.
Lovely. How cool is this? Again, I mean, my mind is blown because you're saying you could take my stem cell and you could, in a petri dish, create a organoid, whether it's brain, whether it will be my brain, with right now. You mentioned there's 2 types of it. One is whole brain organoid, and the second is region specific organoid. We are sitting in 2023. When do you think we could create something which is like really consequential, that we can learn from it so much that we can start thinking about leveraging it to maybe solve some of those really huge neurological disorders that we face?
Let me just back up before answering that particular question. What you had mentioned earlier in terms of, yes, we can take your stem cells to create various types of organoids and tissues in a lab. You know, I think one of the holy grails of regenerative medicine is this idea that all of us have within us the ability to create our own backup cells and tissues, right? So at this point, it's already possible. You can do a blood draw from a person, and from the cells in that blood, you can create pluripotent stem cells. So these are called induced pluripotent stem cells. And from those induced pluripotent stem cells, you should be able to create cells or organoids for any part of the body. And this is not science fiction, to be honest. There are already clinical trials across the world where patients are getting stem cells created from either the blood or a skin biopsy or something like that, creating cells off of them. In this case, the ones I know most well about are for Parkinson's disease. So you create dopamine-producing neurons from these stem cells, this patient's own stem cells, then putting those dopamine-producing cells back into the patient to try to treat their Parkinson's disease. This is not science fiction. This has been done. In this field of regenerative medicine, the whole drive is to see how much further can we push this idea. So answering the question that you pose in terms of when can we start using this technology to start to answer questions, I would say probably right now. It's not something that we have to wait into the future for. It's something that is right now or just right around the corner. One of the ideas of developing, probably the biggest idea behind developing brain organoids is that you can start to really understand neurological disease, especially those diseases that come about when the development of the brain goes wrong. So there are efforts across the world right now where scientists are creating stem cells from patients with genetic disorders that affect the brain. And from those stem cells are growing organoids and comparing them to organoids grown from stem cells of healthy patients, and comparing the two and trying to understand what are the differences, and can you get more insight into the mechanisms of what goes wrong in this disease, and start to think about what are therapies that can develop off of that knowledge. So that is happening now. And there are papers coming out every week, every month, along that line. And so yes, I think we're using this technology now.
Every answer you give me, my mind is actually blown because of the cutting edge of technology of what is happening, and if you extrapolate it maybe 5 years, 10 years down the line, the entire healthcare industry can get completely upended. Today, I think the largest problem we facing as humanity is depression, because of the undue stress we keep on staking, because of the rat race that we are in, to acquire more capital, more wealth, that's like a loop, because the more we do that, the more stress we take. And I think the entire world is getting into that, depressed van. Now, you spoke about regenerative medicine. You mentioned that in us, we have the capability of regenerating, or maybe possibly all of organs in the tissues, but somewhere down the line, possibly we lost it. But there are lots of animals which can still do that. How close are we to possibly, maybe create through this stem cell, induced pluripotent stem cells, whole organs? Because organ transplantation, because it's a huge problem. Right. There's the entire world is waiting. Yeah.
Yeah, so I think that if we're talking about being able to grow a whole organ in the lab, that's still pretty far off. And there are other efforts ongoing right now to see whether or not there might be shorter ways of getting to that goal, of creating more organs that are available for transplantation. So as an example, technologies are being developed where there are animals like a pig or something like that, that is created where they have a specific organ grown in them that is all composed of human cells. So the animal is a pig, but the liver or the kidney is a human liver or kidney. And it's already the case right now that various animal, not so much organs, but anatomical parts of an animal are used in medicine, either as artificial skin or artificial heart valves and things like that. So that already exists, but this takes it to the next level where you're talking about, being able to take an organ from an animal that is human. The cells are human, but it's grown in an animal, and using that. Now, all these things have ethical concerns that you have to think about in terms of the welfare of the animal, and is it a right thing to do? But that from a purely scientific perspective, that is probably going to yield organs sooner than to say completely lab grown. Can we, create these organs? I think that's going to take a lot longer. One of the issues from that perspective is that, and I think this affects not just brain organoids, but all organoid technology, is this issue of blood supply. The organoids that we have right now, they get fed by nutrients and growth factors and things like that in the media that they're the liquid that they're growing in. But that's guided by the physical principles of diffusion. You can only get so far into a tissue by having these molecules diffuse to kind of move into the tissue. And the reason we have blood vessels is so that we can get nutrients and gas and all these other things to much larger pieces of tissue, like our bodies, right? And so, in order to grow larger organoids that look more like organs, I think one of the biggest challenges is to figure out how can we provide nutrients to these larger pieces of tissue without the center of it starting to die. So those challenges will take some time to overcome, which is why I think that lab-grown organs will take some time.
can you explain your research, and the results possibly obtained from the study on transplanting, lab-grown organoids into rats?
Sure. So as you mentioned in your introduction, I'm a neurosurgeon. I'm a clinician. I spend, a part of my time taking care of patients and the other part of my time in the laboratory working on questions of how we develop new treatments for patients. And one of the biggest issues in the clinical neurosciences is that patients can have damage to the brain from a whole different type of reasons. That can be trauma, it can be stroke, it can be a degenerative disease. And the brain does have some ability to restore function. So if a patient has a stroke or brain injury or something else, you can do rehabilitation and patients can get better to some degree, but many times it's incomplete. The numbers I know are more specific for the United States, but between stroke and traumatic brain injury, there are 11 million people in the United States that have problems associated with it, neurological problems associated with it. And there's not much else we can do for those patients. And so, my goal as a clinician is ultimately to try to figure out, can we come up with new treatment strategies to fix these neurological problems in patients with traumatic brain injury, stroke, degenerative disease, all these different things. And to be honest, there's not much out there right now. Like as a clinician, as a surgeon, I can't do too much to help these patients recover. So it's wide open right now in terms of trying to figure out how we can fix these patients. And there's a lot of people that are working on this particular problem because it's such a big problem. And there's a lot of different strategies. One strategy is that we might be able to rewrite the software of the brain, so to speak, by kind of stimulating with electricity to try to get what's still left there to work better in the brain. But it may be the case that some injuries are so extensive or severe that no matter how much you try to rewrite the software code, it's not enough. If you had a computer, you pulled out all these circuit boards and everything like that, at some point, no matter how slick of a computer code that you write, the computer's not going to work. So in those cases, you probably have to put back processors or chips or boards into the computer. And so that's the kind of idea that we're pursuing in the lab right now. If the brain doesn't have enough neurons or processing power, maybe we have to add more back to the brain. And in thinking how best to do this, one of the principles that my lab has been working on is that a tissue with some structure to it might be better than just individual cells that you put in the brain. The brain has such exquisite architecture and structure. And over the many decades of work done by neuroscientists, it's been shown that structure oftentimes informs how the brain itself actually works. And so if we're going to fix the brain as well as we can, we probably have to fix the structure, not just provide more cells, more neurons. And so that's why we were looking at neural tissues and ultimately landed on brain organoids. As we talked about earlier, brain organoids do develop this structure. It's not perfect, but it's probably the best that we have of something that we can create in the brain. You might say, okay, well, the best option in terms of a tissue for transplantation is the brain itself. So can't you just take some piece of a brain from somewhere else and put it into a patient? And that has been tried before. There's quite an extensive list of papers from Europe where they took pieces of brain from a baby rat and put it into an adult rat and showed that, yes, that piece of tissue can become part of the brain. But there's a lot of ethical concerns about that. Where do you get this fetal tissue? So I think that the next best thing is to create something in a lab with structure. And in my mind, again, brain organoids are the best option right now. And so that's why we chose to use brain organoids. And what we were interested in was seeing these brain organoids created from human stem cells. To what degree can they become part of another brain? Now, we're not going to do this in patients right away. So we chose to use an animal model, the standard lab rat to see what happens when you take this organoid and put it into the rat brain. What happens after that?
So would you like to elaborate on what happened? Because you've done the research, you've inserted.
Right. So this took a long time to put all these pieces together, but so just starting at the most basic part, once you put these organoids into the rat brain, and they do survive, and one of the really cool things is that we had these organoids express green fluorescent protein just to tag those cells. And so when we opened things up after we had transplanted the organoids a couple months later, you could use a UV light, a black light, shine it on the brain, and you could see the organoid light up in green. It was pretty cool. And so they survive. And we did a couple of different experiments to show that the organoid neurons were connecting to the rat brain with synapses, the communications, the way that neurons communicate with each other. And so there was a structural connection, but also beyond the structural connection, the organoids were functionally becoming part of the rat brain. So just to explain that a little bit more, in order to show that there was a structural connection, we made use of viruses, and viruses naturally can spread in the brain and cause infections because they can jump from neuron to neuron through these synapses, these clefts that allow neurons to communicate. And so we use these viruses as a scientific tool and showed that if we put the virus into the eye of the animal, you could see that there were connections in the organoid. So the eye had a direct pathway to the organoid, all right? And then to show there was a functional connection, we had the animal was anesthetized, it was asleep, but we had a screen, a computer screen showing different types of visual stimulation, and that was projected onto the animal's eye, which was open. The rats don't really have eyelids that close, so it was open. And we had electrodes in the organoid recording what was happening. And we saw evidence that the organoid was responding specifically to the light. And so that suggested that the organoid had become part of the visual system of the animal.
Wow, how cool is that? So here is this human brain organoid, which you inserted into a rat brain, and it's integrated both functionally as well as structurally.
That's
awesome.
Right.
Now, so talk to me about, when you do this, what are the learnings from this? And through this, what are the brain injuries or the problems that possibly that you'll be able to solve, maybe understanding this?
It's been held for a long time in neuroscience that the adult brain is less what we call plastic. It's less able to adapt than a younger brain. And that's definitely the case for humans. Children that have a problem with their brain from an injury, they're more likely to recover than someone in their 20s and 30s, who is more likely to recover than someone who's in their 60s or 70s, right? So one thing that we saw from our experiment, which was done in adult rats, is that these adult rats, their brains still had enough flexibility that they could accommodate this new tissue, these new neurons. It wasn't so stuck in its ways that it didn't know what to do with these new neurons. And so that is really, that gives me a lot of optimism, because that suggests that we could potentially put new neurons into a patient's brain, an adult patient's brain, and that the adult patient's brain is flexible enough to accept these new neurons. So that's probably one of the biggest things that we learned from this study, is that, it's not just neurotransplantation, putting neurons into a patient's brain. That's not just something for children, but it might work for adults as well. I think that's one of the biggest messages. And in terms of what we can do in the future, I think that the first steps, we're going to be limited a little bit by just the size of the tissue that we can transplant. I was said earlier, the blood supply, things like that. We're still working on that. But kind of the way I'm thinking about this, and I have to give credit to this idea to Dr. Stephen Goldman from the University of Rochester. I met with him very early on in my career, and he brought this idea up to me, is that maybe we can think about these cortical organoids as processing units, processors, right? So one of the interesting things about the cortex is it's thought that there's just many, many of these processors that keep doing the same thing, and these are called cortical columns. There's still a lot of debate and research being done on cortical columns, but it's thought that cortical columns are kind of this basic processing unit of the brain, and it can be kind of used by the brain in different ways. So maybe when we are transplanting these organoids or tissues, what we're doing is putting all these blank processors into the brain, and the brain can use it and say, okay, well, these processors aren't being used for any other function right now. Let me assign it for these different functions. And then the brain can start restoring some of its normal function. And so I think that that's probably the way that we're going to approach this first. It's still ways off in terms of translation, putting this stuff into patients, but that's going to be the idea first. We're going to put in multiple processors, these smaller pieces of tissue into the brain, rather than say we have a huge gap in the brain. We're going to just take this piece of brain that we've grown in lab and put it back in. Maybe one day, but to start, it's going to be a little bit simpler.
What I take from this is that, how everything is connected. That here what you've done is taken a human brain organoid, transplanted into a rat, and it's accepted it and integrated into and thinking that it's possibly its own brain. So how cool is that? Would you like to maybe talk a little bit more about the application part? What are the areas? Because, there are so many, problems. I mean, specifically stemming from the brain, we have Down syndrome, we've got autism, we've got depression, and we've got stroke, and the list is basically on and on. What's the low hanging fruit, which you think that you can, take this out from the lab and into the market?
Right. I think that the initial application of this type of strategy is going to be more for some sort of an injury to a previously working brain. So that is going to be like stroke. That is going to be traumatic injury. I don't think upfront something like this is going to be the answer for autism or an answer for depression or something like that. I think that those problems have other ways that might be more effective in dealing with them. But for something like stroke or traumatic brain injury, where you had a brain that was working to start, and then you lost part of it, some part of the brain got lost, I think that replacing the neurons, replacing the tissues in those cases make a lot of intuitive sense.
What comes next for you? I mean, after doing this transplant on the rat, what comes next? What's the next step?
Yeah, so, this initial study that we did really opens up a whole bunch of other directions that we can consider. One of the biggest things that we don't understand very well right now is what controls, what are the variables that regulate how this integration process works. And the reason that that's important to understand is because if we do understand those different variables, then we might be able to manipulate those variables. And then we might be able to design other strategies to allow that integration process to happen faster. So, fast forward in the future when we can hopefully do this for patients, hopefully we'll be able to say it's not, well, we're going to do this transplant, we're going to put these neurons, these tissues into your brain, and then we're just going to see what happens, and it might take months and months and months, or maybe even years for you to have a benefit. Maybe we can have another strategy to say we're going to do the transplant, but on top of that, we're going to do something else, and you're going to see some sort of a good outcome much sooner, right? So that's why we really want to understand, identify these different factors that might control how the integration happens. The other thing that we are interested in doing is starting to look at, can we put these organoids into the visual cortex, the visual part of the rat's brain. Can we put them into other parts of the brain, and do we see something similar? It's kind of, it is taking perhaps a step back and saying, okay, we need to understand, we have a system that works, but let's understand the system better and know the ins and outs of that system so that we can expand the applications. when we're thinking about translating into patients. And we might be thinking about, do we need to consider other models, other animals that we're doing these transplants and, getting closer to human translation. That's probably a little bit further off, but that is also something that is a possibility.
Are there any negative consequences you see arising? Because, obviously there's the moral brigade, which is always there trying to raise up and say, oh, this is not right. You're trying to transplant a human brain organoid into a rat, and possibly maybe it could be another animal, and so on.
Yeah.
So what are the moral and ethical, possibly those unforeseen consequences maybe that could arise? Is there, you could talk about that?
Yeah, sure. So these are really important questions. Anytime you do something, there's always potentially some unintended consequences in science. We try to do our best to think about these issues and anticipate issues. And we actually published another paper in Cell Stem Cell a couple of years ago, where we talked. The whole point of the paper was talking about the ethics of transplantation. Of organoids and other neural tissues. And we really kind of went into detail, step by step, thinking about this process. The idea that we would somehow make these animals more like a human, I think, is much less likely for a couple reasons. First is that the neurons that we introduced into the rat brain, they weren't spread out throughout the rat brain. They were kind of just focused in the visual cortex. And it's much more likely for neurons spread throughout the brain to have an effect on the brain rather than just one area. The other thing is we put into the visual cortex. And so, what we should be affecting, if anything, would be the vision of the rat. And, this is a bit tongue in cheek, but I'm not sure that our vision is that much better than a rat's vision, And the last thing is we did some calculations in this prior paper to kind of look at comparing the rat brain to the human brain. If we were to replace every neuron in the rat brain with a human neuron, it still may not be the case that that rat is able to think like a human, to talk like a human, or things like that. It's just that there's not enough neurons there to be able to do that. So I think that the likelihood that we are creating a humanized rat, so to speak, where a rat that has human features, is very unlikely. That being said, it is something that we think about, and we do our best to guard against that particular that outcome. Using animals in research, there are many safeguards at this point. There are many committees that review the work that we do, that oversee what's going on, and help us do our best to minimize any kind of discomfort of the patient and make sure that the results are worth the sacrifice of the animals. And so these are always things that we're thinking about actively. And certainly it's not something that we take lightly, that animals are being used for this research.
Really, really appreciate you taking time and being part of the podcast. I think what you're doing could completely possibly upend the way we do healthcare in the next few years. Since you've been invested in the space, you've really been at the forefront of the space. If you had to paint a picture of what regenerative medicine and possibly how brain organoid could play a role in healthcare, the future of healthcare, what would you have to say?
Yeah, I mean, it may be the case at some point in the future that some point early in our life, we have a blood draw that's performed, and stem cells are created, and different organs are generated, and they're kept in some sort of a storage facility where, if we ever have a problem where we need a transplant of this or that, then there is something available to us. If we ever get into a car accident, we have a brain injury, it's like, okay, well, let's stabilize the brain injury, but then we have something that we can kind of order up and fix you with this. That we won't need organ donation will have a supply of our own organs available to us. There's a book by Kazuo Ishiguro, Never Let Me Go, which is really, really interesting because in this particular book, it's talking about people who their only purpose in life is to donate their organs to other people. And so this is a Nobel Prize winning author, and he wrote this very, very interesting book. And that is not my vision for the future. I don't think we'll need something like that because we'll have our own supply of organs. Like it'll be off the shelf. They'll be available to us. Just whenever something happens, we can just order it up, and it will be available to us. And we do not need to wait for someone to come forward as a donor. None of that will have to happen. There's not going to be wait lists for transplanted organs, things like that we have to deal with now. This is just available for you. Now, obviously, you have to think about, okay, who's going to provide the resources for all that? And sure, that is potentially a problem. But what we've seen in science is that things that took a ton of resources a little while ago is much easier now. So sequencing the first human genome took years and years and years and billions of dollars, and now you can do it for a couple $100s, and it's not that big of a deal, right? So who knows what's going to be possible in the future where things become more efficient, and maybe it's the case that generating these stem cells, generating these tissues and cells will become much easier, and truly we will have these off-the-shelf cells and tissues available to all of us.
Thank you, Professor, for taking time and being part of the podcast. I think we're living in a fantastic point of time. The entire healthcare space is going to be completely upended. We're going closer to a point where we could possibly touch human longevity, where we could live longer, not just live longer, but healthier. We are also looking at possibly the opportunity of maybe, like you mentioned, regenerative medicine, maybe, point time where we could just take a specific blood draw and create our own organs. And through these possibly organoids that we'll create, I think, like you mentioned, there won't be a wait line for those people who are waiting for an organ or, a specific neural condition or health condition, and we'll be able to repair and reverse those conditions. So we're living in superbly exciting times, and you're doing a fantastic job. Please keep on doing what you're doing. And to my listeners, if you like what you see in here, then please press the subscribe button. And until next time, see you guys. Bye-bye. Thank you.